without becoming larger, which is called cleavage. So they kind of like become more compact and more dense. After a few days, the cells begin to differentiate. That's when they start to form the types of cells, the nerve, skin, bone cells, etc. The embryo is very vulnerable to alcohol and drugs because the important organs and systems are just starting to develop, so that's why you cannot do any kind of drugs or anything like that when you are pregnant. The embryo is very vulnerable to those type of things, and a common mistake is the fetus develops in the placenta or vagina/stomach, etc., but the fetus develops in the uterus or the womb. All right, so now we turn to genetics, Unit 5. Humans have 46 chromosomes and 23 homologous pairs. Here's what you do have to remember. Humans have 23 chromosomes or 46 pairs of chromosomes or some other incorrect number. These numbers are often confused. You must memorize them correctly. 46 chromosomes, 23 homologous pairs. Thank you. Anyway. So that's what we got. Chromosome pairs carry alleles for the same trait. We all have 2 alleles for the same gene, one from each parent, one on each member of the homologous pair. Sex chromosomes are in humans. Females are XX and males are XY. The Y chromosome is much smaller than the X, so the males carry only a single gene for some traits. This makes males more likely to have some traits like color blindness. These are called sex-linked traits. While genes determine our traits, the environment can affect expression of genes. So if you're in a situation where you live close to the equator, you maybe become more tan than someone who lives in like Antarctica or the Arctic, whatever. Each chromosome has hundreds or thousands of genes to help you build your traits and form who you are, the coding pattern for who you are. Each gene codes for a particular protein, and the common mistake, genes in DNA are made from protein. That's the common mistake. Genes carry instructions to make protein. The genes themselves are made from nucleic acids. DNA is made of 4 bases, A, T, G, C. A 3-letter codon represents a specific amino acid. These amino acids are assembled into proteins. The base pairs, AT and CG in RNA, AU or CG, those are kind of bound together. The AT and CG are the ones that bind together, and T becomes U in RNA, pretty much the only difference. So the RNA carries genetic coding to the ribosomes, and the ribosomes make protein, and that's also referred to sometimes as synthesize. Changes to the DNA are called mutations. They can only be passed on if they occur in reproductive cells, which is the sperm or the egg. That's very important. Common mutagenic agents include radiation, chemicals, and viruses. Mutations may cause a change in the structure of the protein coded for by a gene. This will have an effect on the way proteins work, if it still works at all. You gotta know all cells in the body contain the same genes. Only if some of these genes are turned on, we do not know yet exactly why this happens. So, that's just the very interesting thing. How is it determined what genes turn on and which turn off? So you must know examples of genetic technology. Selective breeding produces animals and plants with desired traits: disease resistance, larger fruit, more meat or milk, or specific colors. Selective breeding makes you, if you win selective breeding, then you got better traits. It's pretty much how it works. The genetic engineering or gene splicing inserts genes of one organism into genes of another. Enzymes are used to cut and copy the DNA segments. Bacteria are often used because they have no nucleus protecting their DNA, and they reproduce very quickly, allowing large amounts of medicine or insulin to be made. The example of gene splicing is something that you must know. The genes to make human insulin was inserted into bacteria. These bacteria can now make insulin that is exactly the same as human insulin. This insulin can be used as by diabetics. This is safer than cow and sheep insulin that have been used in the past. That is a example of gene splicing you must know. New technologies, including karyotyping and DNA fingerprinting, are making it easier to diagnose and treat genetic disease, though we cannot yet cure them entirely. Genetic research have posed many ethical problems that science alone cannot answer. We're going to turn into the political side of things. Unit 6 now, we turn to evolution. Almost done here. Modern species evolved from earlier different species and share a common ancestor. That's what you got to understand. Charles Darwin proposed that natural selection is the mechanism that causes species to change. The basic steps in natural selection are overproduction of offspring. Offspring have variation, competition for limited resources, variation affect outcomes of the competition, survival, and passing on genes or death, and no passing on of genes. So that that's one of the steps of natural selection. Step 4, final step, is variations that are beneficial are passed on, become more common in a population. Those that are harmful become less common because they are not passed on, because the people with the harmful variations will eventually die out. So natural selection, folks. Fit organisms are better adapted to their environment and able to successfully pass on their genes. Unfit organisms usually die and do not pass on their genes, and so their traits are eventually removed from the gene pool. Common mistake: strong organisms are more fit than weak ones. Evolutionary fitness is not physical fitness. Fitness is determined by who is better to adapt to survive in a particular environment and who can pass on their genes. Stronger is not always better. It's how well you do adapt to the environment that you are in. That's what counts. So evolution is usually driven by a change in the environment. This could include a change in the organisms living in the environment, such as a new species moving into the area. Species that cannot adapt to changes in the environment become extinct. Species with little or no variety have a more difficult time adapting to new environments and thus are more likely to become extinct than those species with genetic variety among individuals. A common mistake is the animal could not adapt and it went extinct. Individual organisms die, they cannot go extinct. Only species themselves can become extinct. You got to be very technical with all these tests because you have to make sure that you are putting in everything possible. That's probably the most important thing that you got to remember. Because if you know something and you slip up and you don't put everything that you want to put on the paper and don't express it in the way you want to express it, but you still know it, that would really suck. Anyway, so to evolve, variations must exist in a species before the environment changes, which is pre-adaptation. In common mistake, giraffes get long necks because they need them to eat leaves at the tops of trees. Species do not evolve traits because they need them. Short-necked giraffes were never given long necks. They were just outcompeted by longer-necked giraffes. Better answers are giraffes evolved long necks because the ones with longer necks were better adapted to get food from more than the short-necked giraffes were adapted to get food, or giraffes evolved long necks because more short-necked giraffes died and more long-necked giraffes lived and reproduced. It's a cold world out there. What can I say? Variations exist primarily as the results of sexual reproduction and mutation. Species with more variation are better able to survive the environmental changes than species with little diversity. Gradualism is the idea that says evolution in change occurs slowly. Punctuated equilibrium says evolution can happen in quick bursts. And quick as in not like really quick, but just quicker than thousands of millions of years. Creation of new species usually requires geographic isolation, which eventually results in reproductive isolation. Evidence in support of evolution comes from the fields of geology, fossil record and radioactive dating, genetics, biochemistry, anatomy, and embryology, among others. So some of those are how we can support these and find more about how we and other species, the path of our and other species' evolutions. So classification is how organisms are classified based on their evolutionary relationships. Kingdoms are large groups of related organisms: fungi, bacteria, protists, animals, and plants. Those are the kingdoms we fall under the animal kingdom. If you're wondering, a species is able to successfully reproduce among its members. Branching tree diagrams or cladograms are often used to show evolutionary relationships. This is one where you have two plants, they mark next to each other, and they split out and branch out into change. And it's pretty much on the bottom, it's how much you change, is how far you go out, and then up or down, vertical is, or pretty much along the y-axis is the time gone by. Anyway, we now turn our attention to Unit 7, Ecology. This is going to be one of the most important parts of the test, if not the most important part of the test. You got to understand how organisms interact with their environment, the food webs, nutrient cycles, etc. Energy is needed to keep an ecosystem going. The energy comes from the sun and is made usable by producers. This is plants and other autotrophs. Energy is passed on to other organisms in the form of food. Since all organisms must use energy for their own needs, most energy is lost before it can even be passed to the next step in the food chain. As a result, organisms high in the food chain have less energy available to them and must have, for that point, smaller populations. See the energy pyramid if you want to check that out. This is kind of how that works. So, for example, there's like way more grass, plants, trees out there than there are humans, because humans are at the very bottom of the very top of the energy tree, or one of the top people on the, or top beings on the energy tree, and the producers, the autotrophs, the plants and the trees, are at the very bottom. So they are passing energy along the line until, like, from the rabbits to the deer to the boars, I don't even know, or whatever eats deer that we eat, and then we get that energy. And by the time that happens, the energy is already so far broken down that we get less of it, and therefore we have a smaller population. Even though we still do have a big population, it's nothing near the point where producers are. So anyway, environmental factors, air, water, light, temperature, pH, food, predators, etc. determine which organisms can live in an ecosystem and how large the population can get. The maximum size of a population is called a carrying capacity. There are many roles in the ecosystem, or niche, but competition between species usually results in one species occupying a niche at any one time. Often, organisms with similar needs will divide the resources to reduce competition. For example, birds eat insects during the day, bats eat them at night. So that's kind of an example of how they work together to kind of both satisfy their needs and kind of coexist in a more efficient way. Yeah, so it's also important to know the basic processes of ecological succession. The primary succession is when there's a new civilization, not civilization, but like a new island and a new ecosystem kind of starting from scratch. And a secondary one is one that has to kind of restart after a big disruption in the force, if you will, like a big earthquake. And how is that put back together? So you got to know the following terms: producer, consumer, omnivore, herbivore, carnivore, predator, parasite, habitat, niche, population, community, ecosystem, biosphere, pollution, and renewable resource. Human action, otherwise known as development, industrialization, pollution, farming, overhunting, overgrazing, clear-cutting, introduction of foreign species, soil erosion. Those are pretty much the entire negative human actions that they can do. They often have negative consequences for the ecosystem and for humans too. I mean, we know pollution. There's just some of the things we've been studying recently in bio, for at least my bio class. Negative effects humans have had on the environment are mostly attributed to the increasing human population. Biodiversity refers to the variety of life on Earth. As habitats are lost and species become extinct, biodiversity is reduced. This is considered to be bad because ecosystems with low diversity are less stable than ecosystems with more diversity. Ecosystems with low diversity takes longer to recover from environmental change. We use organisms for many things, such as food and medicine. By reducing biodiversity, we're losing potentially valuable resources. Here are some actions being taken by humans to reduce or repair damage to the environment. They include recycling waste, conserving available resources, using cleaner resources, for example, solar over fossil fuels, protection of habitats and endangered species, use of biological controls instead of pesticides and herbicides that can damage other parts of the ecosystem, just other than what you're trying to kill. I mean, again, even if you're using it and you're just hitting the plants or the pests or the bugs or some of you don't want in your crops, I mean, you're still kind of getting rid of biodiversity. Farming native plants, for example, cocoa in the rainforest. Planting trees to replace those cut down. So for every tree cut down, just plant another one. It's very simple. Rotating crops or planting cover crops to reduce soil loss, passing laws to control pollution, land management, hunting, fishing, etc. Those are all things you can do to help repair or reduce the reduce the damage to the environment. For each of the following ecological problems, you should be able to identify the specific cause and the negative effects on the environment and a way that people are trying to fix the problem. Acid rain could